Bismillahirrahmanirrahim. Today's topic is very important, and that is on research and innovation. And this is Dr. Aftab Ahmed, and I'm founder of Research and Innovation Club. And my email address is given. If you have any further question, I'll be happy to answer. So today we are going to talk about research and innovation and how we can bring innovation through research. As I have mentioned in my previous lectures, that innovation is to bring new combinations, new ideas that can be translated into some tangible products and services and methodologies and so on. So you have a very good idea now that what innovation is and how we can bring the innovation and how we can bring translate the research into innovation. So there are several processes that we will discuss mostly in our universities, as you are a university student. So mostly in our universities we are doing the basic research, and basic research is like we talk about some theoretical consideration and some basic kind of research work, and that creates new ideas. We got lot of more things, like lot of more concepts and other things. So we are involved in basic research. There are several examples of basic research, like we investigate different organisms. For example, if we are going to study the protozoans and how protozoan works, how protozoan work in saline environment, how protozoan works in acidic environment, and so on. So this is some sort of basic research that we do in our universities, and then later on we can translate that. For example, if we talk about some bacteria that can live in some that can live in acidic environment or in basic environment or in heavy metal environment, so how they survive and so on. So that is a basic kind of research work. And then applied research is when we apply those microorganisms into some solutions. For example, if there is a soil that is rich in heavy metals, for example, so in case of applied research, we can put those microorganisms in that soil in a way that they can bioremediate that soil. They remove the toxicants from that soil. So that results in a less toxic environment. So that is one approach. So this is just one example in which you can use the applied research. There are several other ways in which you can use the applied research. And so we start with the basic research. You can directly start with the applied research as well. So some groups are working directly on applied research. For example, they are making vaccines, they are making treatment for cancer, they are making treatment for other infectious diseases. So we can work directly on that. But most of the time, our universities are mainly focusing on the basic research. So we can produce more ideas that can be translated into applied research. And when we start the applied research, that is just the start. Then we have to work on the development work, how we can make the prototype. For example, if we take the example of this bioremediation process, so if we prepare the prototype, then we can actually prepare those microorganisms that we can directly apply into the field. So we start with the development work. Now we have started the practical implication of our research. So then we implement that, just like we started the development, we prepare the prototype, and then we implement that into the field. And if it works there, like if first we do, as you know, in case of applied research, in case of science, we do the lab work. Initially, we start the production in the laboratory. For example, we have to do the bioremediation sort of work. Then we produce the microorganisms in the laboratory. That could be like 1 liter of the culture. And then we prepare some area into the laboratory in which there is a heavy metals. We apply that. And then we check whether the heavy metals are still there or they are reduced. And this is how we can check that. So first, in case of our research work, in case of applied research, we do at the lab scale. Our next target is to do the pilot study. So in that case, we produce more cultures. For example, initially in the laboratory, we have done 1 liter of culture. Then at the pilot scale, we can go for 100 liters, and we can go into a small field and we can do the pilot study. And then we do the industrial level work. So we work thousands of liters are produced, and then we can directly apply into the fields wherever those contaminants are there, and we can. So there is a process we do, like the basic research, as I have mentioned. Then we go towards applied research. Then we start the development work. Then we implement that work, and then we start production, and then we start sale. And from the sale, we we get the revenue. As I have mentioned in my previous lectures, that innovation is that we get some revenue, and the revenue is like more than the business. So that's the entrepreneurship, that if you can get like if from the business you are getting 10 rupees, through entrepreneurship you should get 100 rupees. So this is how you can improve with the. So I'm actually in this lecture, I'm going through most of the cases, I'm going through my own journey into the research work that I have done in Pakistan and in different countries, and how that can be translated into some like applied kind of research, and what opportunities are there, how they can be translated, and what I have done so far in this. Probably I will give one of the lecture about my own startup, that how I started one of the company, and I'm translating the research work that is being done in the universities into the practical field, and how you can also can do similar work. So that could be a totally an independent lecture, that how you can start your own industry, own company or own startup, and how you can take it and take the lead. So I'm going to some of the work that I have done. I started my research work back in 2005 while I was a student in Punjab University. And my initial research work was on the production of hepatitis B vaccine. So my initial work was to express the surface antigen of hepatitis B virus into E. coli expression system. So that was my BS level research work. Later on, when I shifted to another institute for my MPhil studies, so in that case, they asked me that I should go like from E. coli expression system to yeast expression system. So I started my research work in area of Saccharomyces cerevisiae initially, and then I actually moved to United States to get more training and more work in area of yeast expression. Also, I work with a person who is a pioneer in Pichia expression system. So I started working in Saccharomyces cerevisiae expression system, and then I moved into the laboratory in United States in California, where I worked on the Pichia pectoris expression system, in which we use to express the proteins in Pichia system. So here is the difference actually in Pakistan and in most of the developed countries like in United States. So that was back in 2008. And in 2008, still at that time, in Pakistan, the MPhil or MS level work was to clone and express some gene into E. coli expression system or yeast expression system. So if you can clone the gene, and if you can express the gene into the system, that you can get MS or MPhil degree in Pakistan. But and it used to take 1 year of research in Pakistan at that time. So I was lucky to work in one of the institutes that was mainly focusing on the applied research. And the laboratory, they have a very good collaboration with an industry. So in United States, we can express the same protein, like the cloning, the PCR cloning and expression kind of stuff. It's easy to do in E. coli or in other bacteria expression system, but it's a bit hard to express a protein in yeast expression system. So it used to take more time. Like in Pakistan, it used to take more time. But in US, we can do that process simply in 3 to 4 days. We can clone express gene into the yeast expression system just in 3 to 4 days. So it was so fast. And this is how they have actually developed a system that is need to be done in Pakistan. So in that case, if you are like bit familiar with the life sciences, so I will not go into details because some of the students might not be from the life sciences field. So they will not get the idea, but simply it was like that. They have developed a system in which they have used some specific restriction enzyme to cut the gene and put into some specific vector that they have developed into the laboratory. And it was so quick, like you can design the genes primers, and you can get like within 12 hours if you design in the United States, and then you can start the process. And they have the hangers that you have to add into the primers that we used to do, and it was so quick that we can easily clone and express the genes into the yeast expression system. And we used to work for different industry. For example, if some industry is going to make a vaccine, just like there is a challenge now to make a vaccine for the coronaviruses. So in that case, if I'll be working in that institutes, we can do it in 3 days. Like we can get if we know the sequence, we can synthesize the sequence, and then we can put into the bacteria, we clone it and we express it, then we can cut it and put into the PKS system, and then we can express it. So it was so fast to do the that kind of research work there. So we can work like we were working with for the industries. They want to express some therapeutic proteins, and this is my part-time work. I also worked in that company. The company name, the company is still there. It's called Biogrammatics. So I also worked for 6 months for that company, in which we were getting the order to express some specific therapeutic proteins into the PKS system, and then we can get the like expression within a week, and we hand it over the stuff to the company for the large scale production. So this is how we were working. So this is how we can also work in Pakistan. And so I was working on different proteins, like I worked on interferon with thymosin, with albumin. And the thing that I learned in United States is to make the chimeric proteins. So chimeric proteins are actually a combination of different proteins. And that was a good strategy that I could develop in the United States, in which we actually used to express different proteins, like interferon is there. Then we used to put some spacer amino acids in between. Then we express in the thymosin protein. And if like interferon is there, then spacer in between. And we used to express the albumin to increase the life of interferon within the human body. So this is how we were actually playing with different molecules and we used to make the chimeric molecules with different properties, and that was quite interesting kind of work. And we can make like therapeutic proteins. They are like, I was going through some of the injections in Pakistan. They are costing like more than 50,000 rupees or 70,000 rupees, and we can easily do in Pakistan. So there is a lot of work that we can easily do in Pakistan if you start with some of the cancer drugs or some other drugs. We can produce that in Pakistan, and if we like have the whole system that we can develop locally, it's very, so being like the researchers, if you can have the develop in like protein engineering field, then you can ask some of your colleagues to work with you people, and you can work in that area. So that is very interesting work that we initially started. Then I switched to another very fascinating area at that time, and that was the stem cell research. I will not give a lot of details because it's like talking about the stem cells. I have worked for the last 15 years, so like this is the area in which I can talk for hours and hours. But I will give a very brief details about the stem cells. So stem cells are actually the cells in our body which don't have any specific function. So they haven't taken any specific function till now. We can actually push them to make some specialized kind of cells. There are different type of stem cells like totipotent stem cell. That is at the, as you can see, the initial stage of development. So till 8 cell stage, the cells are in the when sperm meet an egg, and till 8 cell stage, they are called the totipotent stem cells because they have the potential to make the whole body. And then there are pluripotent stem cells that are also called embryonic stem cells. They can multiple like in further development process, like at 5 to 7 days. There is outer layer and inner cell mass. So these inner cell mass is called the embryonic stem cells, and these embryonic stem cells can make in our body there are around 250 different type of cells, like our skin cells, our hair cells make the hair cells, like our corneal cells in our eyes, epithelial cells, and so on, and the liver cells and the heart cells and neuronal cells in our brain and so on. So we have 250 different type of cells in our body. Through embryonic stem cells, we can make all different type of cells in our body. So they have a lot of potential, and there is a lot of this area. This is area of actually the stem cell and regenerative medicine, because the research is mainly focused now to cure the disease rather to like treat the disease. So there is more and more research that is going on in this area, and if you are like, if somebody of you is interested in this field, I'll be surely happy to assist you people to know more about the things, how they are working and so on. So this is very fascinating area. And then there are multipotent stem cells. They are the stem cells that can differentiate into limited number of cells. For example, the stem cells in our bone marrow, in our body, there are like in the bone marrow, there are hematopoietic stem cells. They can make the red blood cells, and they can make the white blood cells, and they can make the platelets and so on. So they have the limited potential. In addition, we have the mesenchymal stem cells in our bone marrow. They can differentiate into the adipocytes. They can be differentiated into osteoblasts, the bone cells, and the fat cells, and the cartilage cells, just like we have cartilage in our ears. So they have the limited potential, but this is a fascinating area of research, and there is a lot of research work is being done in this area, and it has enormous potential, because now there is more and more research in this area, and we can cure many diseases through the stem cell research. So this is not only the basic research, but this is also the applied research, and we can cure many diseases through the stem cell technology. Then there is another very fascinating area of research, that was induced pluripotent stem cells. Induced pluripotent stem cells are actually, initially the scientists were thinking that when the cells, they are differentiated into specialized cells, just like our skin cells or neuronal cells, they can't be de-differentiated. They can't be reversed back into the embryonic stem cell like cell. But later on, Professor Yamanaka from Japan, uh, he done some very fascinating experiments, and according to his studies that he has done on the mouse, the mature cells, the skin cells he has initially worked, or the liver cells, they can be de-differentiated. The mature cells, they can be reversed back into embryonic stem cells, like such as I have mentioned, the pluripotent stem cells, and they can be further differentiated into any type of cell. I will give you a quick example, because if we talk, I have worked in this area as well. So if I'll talk, then I'll keep talking about this. But briefly, I would like to add one example here. If, for example, somebody is suffering from diabetes, and is type 1 diabetes, that in that case, the beta cells in the pancreas, they are the damaged. So in that case, what we can do, we can take we can pluck a single hair from the patient, and we can isolate the cells from that, the skin cells, and we can de-differentiate those cells. It takes, like when I was working in United States, used to take 20 days, but now we can de-differentiate cells in 10 days. So we can take the cells from that person, and we can de-differentiate those cells into embryonic stem cells like cells. They have the properties similar to the pluripotent stem cells or embryonic stem cell like cells, and then we can differentiate those embryonic stem cell like cells into the beta cells, and it takes like further 10 to 15 days to differentiate the embryonic stem cells like cells into the beta cells. So we can differentiate those cells into embryonic stem cells, and then we can put those cells into the patient, and we can treat the diabetes. So this is how this works. So this can provide, so what is the advantage of this technology as compared to other technologies? If the same person having the damaged beta cells or no beta cells, if he can get the cells from another person, then first we have to match. Even if it is like there is a lot of there is a matching of the cells, still there is a chance of rejection, or there is a chances that there is a sudden response from the person, and the cells are rejected and not working. But in this case, the cells we are going to take is from the person own cells, and we can take those own cells, and we can differentiate those cells, and we can put the same cells into the person, and we can treat. So these are the person own cells, the corrected cells, and we are putting back into the patient, and this is how we can treat the patient. So this is an amazing technology. I have worked in this area, and still this is in clinical trials. Recently, the clinical trial started in Japan to treat the corneal, the eye diseases, and the results are good. And this is a very fascinating area of research, and it has enormous applications, and it will have enormous application in the future. Initially, the sequencing, this is also very interesting area, because the next generation sequencing is into the market now. Now we have very small gadget that can do the sequencing work. Initially, it was the Human Genome Project that was started in 1990s with more than 2,000 scientists from more than 10 countries, and it was a big project. It has gone for more than 11 to 13 years, and it was a huge project, and spent $3 billion US in that project. So that was a huge project at that time, and the idea was to sequence the human genome. So the initial sequencing, it took it was several countries, more than 2,000 scientists, 13 years, $3 billion US, and now we can do the same sequencing in a day. And now it's cost around $1,000 US, and companies, they are struggling to reduce the cost to $100 US. That is like 15,000 rupees, and you can sequence your genome. So this is how the technology is emerging. and it has it has enormous applications. So we have to do a lot of sequencing, and if you know the sequence, then you can know a lot of things about the human, like uh, the life expectancy, what diseases he can have, how you can manage his life, what food he should eat, and so on. So it has enormous application, and depends like what you want to do. CRISPR is a recent technology. Before this, the genome editing was a challenge, that how we can edit the genome, and it was like a long procedure. It's called uh, thousands of dollars, even millions of dollars sometimes, and it was a very lengthy procedure. But with the CRISPR technology, now we can easily, with the CRISPR-Cas9 genome editing technology, we can easily edit the genome, and there is a lot of hope now for so many diseases that we can actually help those diseases. We can amend the genetic material, and we can treat the many genetic diseases which are very hard, like thalassemia is there, and many other genetic diseases they are there. We can treat those diseases through CRISPR-Cas9 system, and it has not only it has application in human, but it has we can engineer the ecosystem, like with editing, with using this technology, we can get rid of whole species of mosquitoes that can cause a lot of diseases in human. So they are one of the major killers of humans. So we can eradicate the whole species. So this technology is really very powerful. So this is in the form, and this is we can modify the ecosystem, and we can edit different diseases with the help of this. So this has enormous potential, and I have worked on this technology, and this is and I have some collaborations and worked in this area. So this is really good. This is the area I'm going to talk about, the aging, and this is the area I love to work, and I have done for the last 3-4 years. I have a lot of focus in this area, and there are like now there is a lot of work to do the aging research and how we can reverse the aging process. Initially, I mentioned that I have worked on iPS cells. So iPS cells are induced pluripotent stem cells. So that was at the cellular level, we can reverse the aging process. For example, if somebody of your like grandparents, they are 90 years old, then their brain cells will be 90 years old, their eye cells will be 90 years old, although the rest of the cells, they are like changing with the time, but our eye cells, our brain cells, they remain with us. So if somebody is 90 years old, we can reverse their 90 years old cells back into 5 or 6 days old cells. So this is how we can reverse the cells, the old cells, into the very young cells. So this is the reversing of aging process at the cellular level. Now the scientists are thinking, initially, like scientists were thinking that if somebody is old, we can't really reverse the aging process. But now the scientists, they are believing that we can reverse the aging as well. So if there is aging population, we can actually reverse those and make them more young. So there is a lot of research work being going on, especially in area of epigenetics. So we can modify the pattern of DNA, like the methylation pattern and so on. And we can also edit the proteins pattern, like phosphorylation, simulation, and so on. So we can do a lot of editing with these patterning, and we can see what effect they cause. And so there is one component, like it's mentioned there, the resveratrol, that is present in red grapes, and it is believed to be, it is very good to reverse the aging process. And here, one thing I would like to mention is like initially, if you are a biologist, then everything in our body is controlled through the genes. We have around 22,000 plus genes, and there is a family of gene that is discovered in humans that is called the SID-twins. So SID-twins family of gene is involved in the aging process. So we can modify these genes. We can reverse the aging process. So this is also area I can talk. So there is a whole lecture of mine that is available on YouTube. You can go through that lecture, in which I have mentioned the process of aging, the theories, and who you can reverse the aging process. But briefly, in this lecture, because the time is short, so I will just like to mention that there is a family of genes which are known to be involved in the aging process. I have also I'm working in this area. So we are trying to reverse the aging process. And there is one very fascinating slide you can see over here. There is one compound that is known to you probably, that is NAD. And so NAD is just to give you an example that NAD is present in human cells because it's involved in the mitochondria. The electron transportation. NAD is converted into NADH, and it's involved in electron transport chain and ATP production. There is a reduction in NAD level in the human body. So, if, for example, at the when we born, then it's 100%. At the teenagers, it's reduced to 50%, and when we reach to 50 years old, it's further reduced to 50%. So our energy in the cells is further reduced. So what happens? Scientists are now thinking that if we can restore the level of NAD in human body, then what we can do. So that was a very very fascinating work, and that was done in Howard University, in which they fed the mouse with NMN, that is a precursor for NAD, and when they fed for 2 weeks, then the 2 years old mouse that were reversed, the aging process reversed, their blood parameters changed, their physical appearance changed. And that was that was equivalent to a 6 months old mouse. So that was, you can't believe that how fascinating is, like, if you can reverse the old into something very young. So that's just like if there is a 60 years old person and you reverse that into a 20 years old young man. That is like, this is the link of the research article in which I have taken these images. I have worked on this NMN. I got the chemicals from one of the company in the United States, and we have done the research work that is in the process of application. But we got very good results, and especially we have worked on the skin cells and the brain cells and how we can reverse the aging process in these cells. And we also started working on the organismic level, that how we can reverse the aging. So you can imagine from here that if you can find out a single molecule that can reverse the aging process, how big is in that? This is a billions of dollars industry, and you like the total export of Pakistan is right now is around 19 or 20 billion US dollar. If you can make some compound, I can surely say that you can have more than the total exports of Pakistan. So this is how the fascinating is the innovative research work, so that you can do. So this is a really amazing thing that we can also do in Pakistan, and you can also think like how you can reverse that. This is another area that I have worked for my postdoctoral work at Nara Institute of Science and Technology in Japan is related to tissue engineering. This is also an area very fascinating, and I have worked some like this we can also do in Pakistan. And tissue and organ damage is a worldwide problem. There is a shortage of liver, there is a shortage of kidneys, and if you have to get the organ from somebody, it's very expensive, and it's also very hard actually procedure to get, like, convince somebody to give you the kidney or convince somebody to give you the liver. There is in Pakistan, due to hepatitis, there is a shortage of liver donors, and due to high percentage of chronic kidney diseases, there is a lot of shortage of kidneys. So this is a big challenge. And what could be the solution? One solution is we use the artificial prosthesis. We can use the mechanical devices, surgery, drugs, cellular therapy, transplantation. These are the solutions that we can try to these patients, but these are sometimes not the long-term solution. So what we can do, we can go towards the tissue engineering, and in tissue engineering, we can work on the natural scaffold. We can work on the artificial scaffold. I will give you some of the example of these, and we can also go for bioprinting. So the technology is very simple. If, for example, if there is a damage in the knee, there is a common problem now in Pakistan due to the osteoarthritis. So we can get the cells from the same patient. We can prepare the scaffold. That could be a natural scaffold. That could be an artificial scaffold. We can put the person's own cells into that scaffold. We can grow it, and then we can heal the damage. So, for example, if there is osteoarthritis and there is a no cartilage into the knee, so we can get the cells, we can prepare a scaffold, we can develop the cartilage into the laboratory, and then we can repair the knee. And this, there is a lot of work is going on in bioprinting, and our skin is successfully printed by the bioprinter. So this is another option that we can use Natural scaffold, I have worked on a natural scaffold. So natural scaffold, they have a lot of advantage. For example, they are the biocompatible because we are using the cells from the patient, so there is no immune system activation or drug required, and we can regain the tissue function, and we are not using uh, the embryonic stem cells, so there is no controversy. So this technology is very simple, as I mentioned. For example, if there is damage into the lungs, just like COVID is damaging the lung, so what we can do, this is, for example, in rat lung, so we can decellularize it. So through the decellularization process, actually what happens, we can only remove the cells, but their extra cellular material remains intact. So we use the approach in a way that we can only remove the cells but not the extra cells. So it's just like, you can take the analogy of this wall, for example. So in this wall, if we can remove all the bricks but we don't remove the cement in between, so you can see that all the bricks will be gone but the cement will be there, so we can put the new bricks in that and we can make the walls again. So this is just like that. So we can reseed the lung and we can prepare the lungs, and then we can engineer the lung, and then we can put into the patient. So this is one of the solutions. This is a lady that was treated in UK, and there was actually many scientists from different countries like Spain and other countries. They worked together to make the human trachea. So she lost the trachea due to infection, and after that, the scientists, they took the cells from the same lady and they made the scaffold, and they grew the trachea and put in the, and she's living fine. So this is a very like fascinating area in which you can treat. Professor Anthony Atala is a very famous tissue engineering person working with Wake Forest Institute, and he has worked on several tissues. For example, he has worked in human bladder. It's very simple to create the human bladder because it's not a complex organ, just like liver or heart. So in this case, they took the cells from the patient, and they took they grew actually they took the extracellular matrix, and then they grow the cells, and then they transplant it into several patients, and it's working fine. So this is a technology that can be easily applied. So the technology is very simple. These are you have to prepare a cocktail of different chemical and enzymes, mostly like 1% PEG, 1% Triton X100, and 1% STS, and we can easily decellularize the tissue in which we remove the cells but not the extracellular matrix. And this is one of the studies that I have done in Japan. The decellularization, so liver problem is very common in Pakistan, and we can grow the piece of the liver in the laboratory, and they have enormous applications. So this is the liver that I got after the surgery, and then I started the decellularization process, and you can see the extracellular matrix is there. Only the cells are being removed, and all the veins are still intact. So this is the final form. You can see the liver is beautiful liver. All the extracellular matrix is still intact there, and it's very beautiful. So what we can do, we take the liver, we can decellularize it, and then we can reseed. So in this case, I took the GFP positive cells, so I can see whether they are growing in the laboratory. So we can take the tissue, and we can re-inject the cells, whatever, like if we have to grow the uh, uh, liver, then we put the hepatocytes, and we can put the hepatocytes, and we can see whether they can make once again the liver, and we can put into the patient. Similarly, I worked on that heart, and I took the heart, decellularized it, and then I reseeded the cells of the heart cells, and you can get in like 10 days, you can get a beating heart in the laboratory. So this is how fascinating it to look at the beating heart in the laboratory. But if we go towards the transplantation, yet it has to do a lot of more work because heart is a very complex organ, and to get the same pumping capacity is really a challenge. But for the simple organs, for example, if we talk about the skin in Pakistan, so in Pakistan we get very expensive skin from different countries. I have worked with different hospitals, like in Islamabad. I have gone to the PIMS burn centers and other hospitals, and they get very expensive skin grafts. We can develop the skin grafts in Pakistan, and I have started one of the project in that, and my like skin that I have developed in Pakistan, that is in the clinical trial. So after it, if it works, then we can actually translate. So in the future, we can make any type of organ in the laboratory. If we like somebody, we know that he has a kidney problem and kidney is being damaged, we can get the cells from the person. Like he's going to have a problem after maybe a year or after 6 months, we can grow the kidney into the laboratory and the person can. Or if somebody has a liver problem, we can grow the liver in like in 2 months, and he can give the sample in advance and can go into the laboratory and the person get the piece of the liver and go to the surgeon and they can rather like we are getting from a donor. That is a very risky procedure to open the whole chest and get a piece of the liver and then close it. And several cases they died, the donor died because of the infection after the surgery. So this is a very fascinating area. I have worked on one of the project that is very interesting. Time is really short, but just to give a very quick overview, optogenetics is a field in which we control the uh, neuronal networks through the light. So we put the specific wavelength of light in the brain and we control brain function. So actually what happens, just like the photoreceptors, they open to the light and they take the image from the light. Similarly, there are several proteins like the photo, just like we have the photoreceptors. There are proteins like channel rhodopsin and archaeorhodopsin. They are present in bacteria and archaea and algae, so we can get those proteins and we can express those protein into the neurons and we can activate neurons or we can put into the silent form. So we can generate the action potential or we can lose the action. Our brain functions, they are controlled in specific region like our sleep, our appetite, our anger, our depression. So everything is in our brain. So for example, if somebody is depressed, then we have to get uh, the person into the medicines and he has to get the medicine for months to be a normal person and it has a lot of side effects. So in this case, if actually it's through the pleasure center that is in our the brain, and if we can activate the pleasure center, the person could be normal. And drugs takes a lot of time to be action, and this is approach that takes milliseconds. So if we put some specific wavelength of light into the brain and we can activate it. So for example, if somebody is depressed and through this approach we can put specific wavelength of light into his brain and we can activate the center, and in millisecond the person will be normal and the depression will be gone. So how powerful this thing. I will give more details about this. So entrepreneurship, if we talk about HP, about Google, about Microsoft, about Apple, if we talk about all these giants, just like Google or Microsoft or Apple, Apple has the assets are more than 400 billion US dollars, and Pakistan normally have around 15 billion US dollars reserve money. So you know the difference about these big giants. But they started from garages. So if you have the spark, just like you have taking this course, you definitely have the spark. You can start the work in your house at some specific location, and you can start working and can work on the prototype and if test the prototype, and if it works then you can make some collaboration with others. So you can get the money from your family or friends or fools just like the three f they give in the business term. So you can get the money and you can translate your idea into some tangible product. So this is how they have started. You can also do the same and we can do the same in Pakistan. And our idea for this course is to push you and to motivate you and to motivate ourselves as well to take the start, and we can do some big impact in the world through our services. Engineer Aslam Azad is a very good example. I often give his example because he started with his idea, started producing cars that are solar powered, and he has made several cars, and the golf cars are shuttle services cars and vans. And initially started in Sheikhupura. Initially he used to produce a car per day, but now he has another factory close to Islamabad, and they have more production capacity now in Pakistan, and they are so he has taken a start and he's translating this just like we can. There are hundreds and hundreds of examples in Pakistan that we can actually follow. So the take-home message with this lecture is to share your ideas with others, with those people to whom you have the confidence, so they can help you to polish your ideas and help you to translate and share your resources with each other, so you can help each other to grow. Have discussion, because discussion, if you have the discussion with the positive people, they really help you to narrow down your idea into some action that you can translate. And have a very good networking with others. Networks really help you to grow. I was in United States for the first my visit in 2008, and I was in one of the lecture, and the person mentioned that 70% of your success in your future life or the success of your business depends on your network. So always try to have good networking with the people who are successful, so they can help you to be successful. Have good collaborations with others, share the things, have good collaborations, learn from others, the things you can't do, ask the other to do for you, so that you can do through the collaborations. And if you can do something, you are definitely inspiring others. So keep inspiring others, keep evolving. Keep evolving means try to learn something new every day, so you can grow yourself, so you can grow mentally, you can grow economically in every perspective. Sustainability is very important. We should keep forward, but we should work also on the sustainability. And you are presently in the university, so they have the best places, as I mentioned in one of the lectures. There you have mentors, there you have resources. If you have the idea, you can work, you have the freedom of work, you can do and try to translate your ideas into something tangible. So the assignment for this week is for the last lecture you have gone through on the health technologies and innovation and this research innovation. You have to, the assignment is one innovative research topic. So you have to send to us. You have worked on one innovative research topic. You have prepared one topic that you think could be innovative, and you can also work on that, or in future you can work on that. So you can think about some topic and you have to send that to us. With this, thank you very much for your listening. If you have any question in these topics, so each topic is like we can explain. These are some of the examples. There are many other ideas that can easily, just like I can give you. Yesterday I was I need some yeast extract, and I went to the market in Islamabad and Rawalpindi, and I went to several scientific stores, but I couldn't get the yeast extract because they haven't supply here. And they said that they will get from Lahore or Karachi, and it was also very expensive. So I was going through the methods in which we can produce the yeast extract, and I was thinking that you can easily do it. So in Pakistan, there is a need of yeast extract. Now, India is making it. One of you could be making it. So in each thing, in the innovative research is like you have to work on some innovative idea, some innovative approach, and we can work on that. And this is how we can actually grow. I have visited some several industries in which they have mentioned that we are following this process, but there is a way. If we, for example, they have the heat method, and if we go to enzyme method, they can reduce the time and cost and everything. This is the innovation. For example, if they are making from A to C, and the cost is 1,000 rupees, and it takes 3 days. And if we can go to A to C through using some enzyme, and the cost is 100 rupees, and it saves time, it's innovation. This is how we have to think that what contribution we can do. And what ideas we can work on. This is just to give you an idea that these are the areas. There are hundreds and thousands and millions of areas in which we can work. Each thing we can contribute if you think innovatively. So you can contribute definitely, and we should contribute. And this is how we can make an impact. Hopefully, you will work on the assignment, and we should get it before the next week lectures. And lectures are going to end very soon, so you also have to work on your projects, and you should start working on that. If you have any question, you're most welcome to ask through email or through the WhatsApp group. Thank you very much.